So what really is pulse field ablation? It is a field-based approach to cardiac ablation. It involves the use of pulsed electrical fields sustained around an electrode or an electrode array to ablate cardiac tissue. The mechanism of cell death that PFA uses is called irreversible electroporation. When cells are exposed to high electrical field gradients, this induces a permanent hyperpermeabilization of the cell membrane, which leads to cell death. Here's an example demonstrating the transient pore formation in the cell membrane just nanoseconds after the electrical field is applied. Now, how does this work? Well, nanoseconds after pulse delivery, temporary pores are formed in the cell membranes, making the membranes hyperpermeable and disrupting the cellular steady state, otherwise called homeostasis. Within seconds to minutes, the hyperpermeable cell membranes enable ions and molecules that are normally not permeable to either flow into the cell, like calcium, or exit the cells, like ATP and other high-energy phosphates. Within minutes to hours, irreversibly electroporated cells undergo cell death via a variety of mechanisms, including programmed cell death, otherwise called apoptosis. However, the series of events that lead to cell death of cardiac tissues likely involve a range of cell death mechanisms. For example, apoptosis, or programmed cell death, is one of these such things. But why would we care? Well, the timing nor the specific cell death mechanisms in PFA are not fully elucidated yet, and may, amongst other things, also depend on the pulse waveform and on the catheter delivering the waveform. What we do know is that PFA results in replacement fibrosis or scar chronically that is similar to other ablation technologies. So how long does it take for cells to fibrose and make a mature lesion with PFA? While cell death may occur within minutes to hours, it actually takes much longer for the lesion to fully form and stabilize, with some data suggesting that it could take 7 to 10 days. This is important since we may not know the final results of our ablation until 1 to 2 weeks later. It is unclear if this can allow us to further change the acceptable blanking period after ablation, and further clinical data on this is required. For example, in a sub-analysis of the PULSED AF trial, we showed that if early recurrence happened, it usually occurred at 7.5 to 20 days post-ablation. Almost all the early recurrences occurred within the first month.